Yeah, you can pass it the next one. Not sure if anyone else can join. We'll wait 2 more minutes.
I choose it. I just read the abstract. I thought it was important also for my research.
Yeah.
Uh-huh.
Oh, are you
That's
so far
Uh-huh.
Are you gonna get some eagle jumping paper?
No, we have a lab meeting today and we have.
Another paper?
We have another paper we have to discuss for our meeting and we were just saying, we either of us have read it yet, when the meeting's at 1.
What will
There was another paper for this class
for today?
for the Think and React.
Yeah, that wasn't
suppose
posted until like 2 days later.
Yeah, that's fine. And then there was the other Think and React that looked like it was due over spring break. Cause it was posted like the Friday. I don't know. I
choose the
papers to read.
I did it in place of the one that was supposed to be posted, and then I saw the other one, I was like, heck.
But those doesn't have dates.
No, it's kind of like just.
Do it before the year ends.
No, I think it's okay because the one last Wednesday is not due till this week, right? And then you're gonna have 2 this week, but they're not gonna be due till later, so even if we post it a little bit later, you still have a little bit of time, and we talked about it, so even if it wasn't posted, we knew where to find it. And I have the link in the, oh, I guess, yeah, the lecture.
It was a very
posted.
nice paper, though. I actually recognized the person who conducted the research. It was the same one from that Ant Lab video with, it was literally one of the researchers was the same guy who has the who does the Ant Lab YouTube channel. Like, it was literally the exact same research. He published a video about it in August of 2021, and then here's the real paper, as of January 2022.
Interesting, but having that delay is not common. Normally it's the paper comes first and then you do a YouTube video to sum it up.
Well, I think it was because technically the paper was like in the pre-publishing phase, so we could still technically talk about it. So it was like,
There's still things that can happen though, at like pre-publishing, like you could feel like, yeah, we're just not doing this anymore.
I mean, it was effectively done. Even some of the images that appear in the video, like the CT scan of the larva and some of the graphs that were used in the paper. So it was kind of like a summary of the paper before the paper even came out.
Okay, we're gonna start. I hope you can hear me, Kyla. Okay, let's start, and if other people come in, then I add them to the group. Okay, so we're in that section that encompasses locomotion, communication, and behavior. So it's the second lecture in that section about communication. Why is it so slow? Okay. So what are the roles of locomotion, of communication? Sorry, I don't know, I'm stuck on that last week. So there's intraspecific communication that's used mostly for mating, alarm, competition, for resources. Interspecific communication, and I focus on that that's what the article focuses on, and we'll get back to that. And then there's communication with the environment, but all of these types of communication were selected by evolution to ensure survival and reproductive success for some of the individuals involved in that communication. Okay, so like I mentioned, intraspecific communication, that's within one species, and so that allows to identify who is a potential mate, mainly. So it saves energy that would be wasted on mating attempts that would be unsuccessful, like mating with objects in the environment or with a completely different species, obviously that wouldn't work. So having this system of communication to really recognize who is a potential mate is very helpful for animals in general. Then we also have competition for resources within a species. So the communication messages could be for cooperation, like we're all going to work together to get food, or avoidance, like there is already an individual of my species eating that, and sometimes it's better to avoid rather than confront. And so the other type of interaction here would be chase the competitor away. And so that's when, instead of avoiding, they will fight. Then we have interspecific communication, so between species. And one of the subcategory that we're gonna review a little bit is protection from predation. That's pretty important, obviously. And that's how defensive communication has evolved through selection of the individuals that escaped predation. Then again, we have competition for resources. You're not just competing with your own species, it's with a bunch of different other species. It could be just insects, arthropods, animals. It has no limit as long as these individuals feed on the same thing or use the same area. And then I added communication type that involves 3 individuals, and we're going to get back to that one too. It's between the herbivore that attacks a plant, but the plant sends a message for parasitoid to come help, basically. Again, just to be clear, what is communication? There are a few different definitions, but some of them are not really satisfactory. For instance, there's an aspect that involves increasing fitness of all the participants, and that's kind of part that we're gonna change. Just a second. But so basically, communication is a codified exchange of information between 2 individuals, same species, different species, and it was forged through evolution, natural selection. And the reception and interpretation of the code that could be visual, auditory, chemical, tactile, must be beneficial for both. That's where we don't agree on that, because imagine like you talk to someone, you argue, then at least one of you is not going to be happy with the outcome. So is it not communication then? So I don't know if we can keep that part mutually beneficial. But so it results in the increased fitness of the participants, either one or both, or if there's more than 2, an entire group. But so for this class, we're going to consider that insect communication in its broadest sense is when one individual gives off a signal of any shape and form that produces a change in behavior in another individual. And let's keep it at that. So there are several ways that insects can communicate. Some of them are pretty obvious: visual communication, acoustical communication, which would be the equivalent of hearing for vertebrates, tactile communication, and chemical communication. And they are classified this way depending on what receptors are involved in receiving the signal. And they all have specific characteristics in terms of the range, the rate of signal change, how fast it can change, circumventing obstacles, and how much it costs energetically for the individual that sends it or receives it. And so for visual, for instance, the range is medium. It really depends on how far, what is the capacity of the eye of the insect that we're talking about. So medium is kind of a middle ground. But also you can relate that to yourselves. Like you can't see past a certain point, and it's the same for insects. The signal change rate is pretty fast. But if there are obstacles, then you can see through an obstacle, and it's the same thing for insects, and the cost is pretty low. But if we compare to chemical communication, there is a longer range because the molecules produced can travel in the air. The rate of signal change is slow because it takes a whole process to regulate the production and the emission of a chemical in the air. And then once some of them are very persistent, so it takes a little bit of time to change the message there. The circumvention of obstacle is pretty good because it doesn't matter if there's a big rock, then the chemicals can go around it, above it. And the energy cost is relatively low. And so for the other two, there's just different characteristics. And for a message to be transmitted, there's sometimes combination of different stimuli. Okay, let's start with visual communication. So usually it is used to identify food and mates, and also to orient insects, to orient themselves in the environment. And they use their compound eyes, which have high resolution, to identify ritualized and stereotyped behaviors or behavioral sequences. And there are two examples. Hymenopterans are really good at recognizing faces among their family hive. And they can also, at least wasps for sure, can recognize abdominal structures like yellow patterns. And so that's how they can recognize if an individual belongs to their group or if it's an outsider. And so that's because hymenopterans can perceive different colors that are in they're using 3 spectral photoreceptors, and they peak at 340, 440, and 540 nanometers. And so here's a representation of the range. And for comparison, this is what humans can see. So hymenopterans can see in the UV, and we can't, but then we have this common area, but they also appear not to be able to see red very well. In butterflies, there's been a diversification of the photoreceptors that allow them to see more colors. And I don't have a graph, but for swallowtails, at least they can see 8 different spectral types, comparing to the 3. And so they can also perceive long and violet wavelengths. And also, I just wanted to mention, sometimes I put DOI of papers that I thought were interesting. If you have questions, you can always refer back to that for the specific research I mentioned. So then visual tracking is used for identification and tracking of food. And it's more specifically for predatory insects, because they use the visual signals sent by the prey to track it and get it, basically. And we're going to talk a little bit about the praying mantis, because that's the best understood insect that uses visual tracking. And it might be the best understood just because of its size. It's easier to observe than little tiny flies or wasps. And most of you have seen one of those Chinese praying mantises, and they're pretty big, so maybe they're just easier to study because of their size. They also have pretty big compound eyes. I have 2 of them, and they can assess the distance of the prey on the background based on triangulation, so they can kind of zero in on their prey using their binocular vision. And it works at a relatively close range too, because they have to take into account the size of their raptorial legs. They want to be able to grab the prey. And so it's about 25 millimeters, 2.5 centimeters. They also have a relatively small distance between their eyes, and so that kind of limits their range. So they're limited based on the size of their legs and the range of their eyes. They can also move their head by saccadic tracking to maintain the image in the foveal region of their eyes. And yeah, that part I'm gonna have to read, I'm sorry. So that provides high spatial resolution. In saccades, I'm sorry, that's just the kind of word that I've only ever read and never heard it, so I don't really know how to say it, but there are rapid head movements. And so, you know, when you move your head very fast, the image is kind of blurry, and so they have a way to really zero in on their prey and stare at their prey to make the image stable. Either the head movement or the eye movement, very fast eye movement, can make the image blurry, so the insects, predatorial insects, stare to make the image fixed. And this is the structure of the compound eye. I'm not going to get back to that, but I'm pretty sure you've already seen that already in the nervous system. Okay, and I mentioned the visual defense at the beginning of the lecture. That's one of the strategies used by some insects to avoid predation, and that uses patterns, visual patterns that are recognizable by other animals. And I have two examples. So the first one is this butterfly that has a pretty camouflaged underside, and so it will keep its wings up. But then if it sees a danger, a potential predator, it can lower its wings, and you can see here this resembles the eyes of a vertebrate, and so that is supposed to scare a predator away. And so for the second example, it's kind of the same strategy, hiding and also scaring the predator away, but it uses the wings differently. The top wings are, the forewings are camouflage based, and the bottom wings are what is supposed to startle the predator. And so when it unfolds the wings, that's when there's this bright color flashing at the face of the predator, and it's supposed to be startled, run away, or releasing the mob if it's already in its mouth. And so, yeah, that's a passive defense mechanism. And I wanted to talk about quickly this strategy that's used with by the pea aphid. There's been evolution of two different morphs. One of them is green. It's the wild type, if you want. And there's a reddish morph. It's not exactly bright red, but it's more towards the red. And it might be because parasitoid wasps can see better green aphids on a plant, even if it's supposed to be camouflaged on the plant, but they evolve that way to focus on the green aphid, and so they can see it pretty well. But the morph evolved through a symbiosis with a fungus that allows them to use pigments that are produced by the fungus and change their color to a more reddish color, and the wasp is unable or not as able to see them. And that kind of goes back to what we saw here, if we believe this graph, and we do, obviously. This is for the honeybee, but there's going to be a lot of similarity with other hymenopterans. And as you can see, they are not, they do not see very well in the red. So that would be an explanation of why they can see the green aphid but not the red one. Okay, another type of communication that goes through vision is bioluminescence. And it's used to be able to be seen by a member of the opposite sex of the same species. So it's for mating primarily. And that probably evolved because, to find a mate during the day, you would, if you were an insect, you would make yourself more visible to predators because they are more active during the day. And especially in the environmental background, if you move, then that makes you very conspicuous. So one solution is to communicate at night with your potential mates, because the predators are less active in theory at night. And so a way to do that is to produce light to be seen by your potential mates by bioluminescence. So it's not clear exactly how it evolved. It could be a new process, or it could be side effect of another process. Basically, that's a way to explain that. It could be incidental to another biochemical pathway. And I mean, that has to do with the fact that the oxidation of luciferin produces a lot of active oxygen, which can be detrimental, could be toxic to the insect itself if it accumulates too much. So there must be a way to use that oxygen, and that would be an antioxidant effect. So there's a lot going on there. And there's an example of another function of bioluminescence where it is used rather as a defense than for mating. It could be used for either luring a prey or in terms of defense as an aposematic signaling. So aposematic, it's a signaling to a predator that you're not good to eat. And so that would be the case, for example, that's what they found out about collembolans. That's not a great picture, but it's just to also show you the patterns of bioluminescence on these arthropods. So yeah, they use bioluminescence for another function than mating. So it's not exactly sure how that came up and what was the primary function. But I was a little confused, I'm sorry. But it evolved in at least 9 families within 5 groups of insects. So we have the Collembola that I just showed you, Homoptera, Diptera, Dictyoptera, and Coleoptera, which is what we know best because there's just more beetles in general that show bioluminescence. And so the main 2 families that show bioluminescence are Elateridae, the click beetles on the left, and Lampyridae, which are the fireflies that I'm sure you're all familiar with, especially in the Midwest. Yeah, and you can also see that there are different structures involved in these 2 different families. So if we talk a little bit more about fireflies, they produce a yellow greenish light, and the wavelength is between 550 and 580 nanometers. And their light organ, that's called a lantern. We're going to get back to that right now. I'm blanking, but I think that's a lantern. It's derived from fat body on the ventral tip of their abdomen. So if you have a firefly belly up, it's on the upper side. And it's used to identify potential mates, but also to light up potential landing sites, or their landing sites, potential, they're right there. So different species of fireflies use different strategies. Either the female produces the light to attract a male, or males produce a flash pattern, and females nearby that would be available return the signal. And so they use different patterns to, or different patterns are specific to a certain species, and it's used as an isolating mechanism. If there are 2 species overlapping, it's one flash pattern for one species, another one for the other, and so they recognize that they are not compatible. And so the way that works, how they initiate that light, that bioluminescence, they can perceive a decrease in light outside. So when it's close to nighttime, and then they can start flashing at that point. Sorry, like I said, they emit in green and yellow because the eyes of the receivers are adapted to these colors. Basically, that's what this means. There's an example of the Photinus firefly. So the male patrols to find females. It flashes every 6 seconds. Then it pauses for 2 seconds to wait for the response of a female nearby. And then it flies near the female and then engage in other exchanges. And one other species of fireflies has evolved a strategy that recognizes the flash pattern of the Photinus males, and it's able to mimic the response of the female of their species, of the Photinus species. And the males are then attracted, they come closer, but these are predatory fireflies and they feed on the males. And so that's what we call aggressive mimicry that lure and prey the males of the Photinus genus. Just to mention how close those two names are. So Photinus are the prey and Photurus are the predators. Okay, and the physiological process. So first the structures, okay, so I was right, it's called the lantern. The fat body cells constitute the lantern. And so the photocytes are the cells that are responsible for light emission. They are right beneath transparent cuticle, which makes sense because the other fireflies need to be able to see it. So the cuticle is very thin and transparent there. There's a lot of photocyte granules and mitochondria. There's tracheoles. So it's like we talked about last week with the locomotion system. When you need a lot of energy or oxygen, you have mitochondria, air coming through breathing. So tracheoles are part of the respiration system. So you can have an idea that there's going to be a role of oxygen, energy, and air from breathing. The photocytes are arranged in that circular pattern, and they're separated by cylinders that contain nerves and tracheal bridges. And the nerve innervates the tracheolar cell, not the photocyte. And then on the dorsal side, so again, if you take that firefly belly up, the dorsal side would be more towards the inside. It has a layer of white urate crystals that serves as a reflective surface to make the light really pop. So basically what happens to initiate a flashing, to initiate bioluminescence in fireflies, they receive the signal that the light is going down in the environment, a nerve impulse is generated, and it initiates the pattern. But there's also a role of oxygen in this. So first, the nerve is stimulates the tracheoles, which causes the uptake of tracheal fluid and oxygenation of photocytes. Then nitric oxide gas is produced in the cells between synapses and tracheal cells, which will inhibit mitochondrial respiration, which increases oxygen levels and allow oxygen flow into the photocyte. So if you remember in the muscle, it was kind of the other way around. They need a lot of oxygen to produce a lot of ATP. So there's a lot of energy consumed during a contraction. Here it's kind of, not that it doesn't use energy, it's just that the mitochondrial respiration uses a lot of oxygen, but we're going to need the oxygen for another function. So that's why the nitric oxide gas is used to inhibit this respiration. so the oxygen can be used another way. So like I said, the nerve impulse initiates the flashing pattern, but then the oxygen is what controls the flashing. Again, this is the process of how luciferin is converted to produce light. It goes through different steps, and the enzyme that allows it is called the luciferase. So it's pretty easy to remember luciferin, luciferase. The luciferase oxidizes luciferin, which produces oxyluciferin, oxidation, oxy, and you just add that to the name of the molecule. That's pretty straightforward. And that's what yields a photon of visible light. So luciferin is, brace yourselves, a benzothiazole derivative. And a lot of species have the same, or all the species have the same molecule, but then what changes is the form of the luciferase, the isoform in the different species. And so they can break down luciferin, the different versions of luciferins by the different versions of luciferase, and have different type of light in the end, different color light, slightly different. Yeah. Let's keep that because I'm running a little late. If you have questions, we can come back to it. So we're going to move on to the acoustic communication. Insects produce sounds and vibrations, which we consider acoustic, through the air, water, solid substrates, and they can receive and interpret those signals by what we call hearing, but that they don't have ears like we do, so it's not exactly the same thing, but that's the closest that we can compare it to. And so the vibrations can be produced by different mechanisms and through different types of substrates. It's been described in at least Orthoptera, Diptera, Coleoptera, Lepidoptera, and Homoptera. It's not clear if other groups also use acoustic communication. It's not that it doesn't exist, it's just that it hasn't been described. And so here is an example of the face of Anopheles gambiae male. And that is the organ that receives vibration. That's the Johnston organ. And that's just one example of what type of insects receive or emits and receive acoustic communication messages. So sounds can be produced by percussion. That's really hitting something and making noise. And so, for example, the forewings of some insects are turned into or modified into small knobs that are called castanets. And I think that is because castanets are musical instruments that you hit against each other and they produce percussion sound. And so when they strike each other, they produce a sound that resonates through the wings. For example, in this southern whistling moth, there's another example of an insect that produces sounds through percussion by striking a body part against a substrate. So Plecoptera, stoneflies, can drum a substrate with their abdomen to attract mates. Then slightly different sounds by vibration. So in this case, they use a muscle or muscles to directly vibrate a membrane. For example, the timbal that we can find in cicadas, and it's also found in some moths like arctiids and noctuids. How does that work in cicada? So basically, it works like the lid of a tin can when pressed and then released by a finger. So on the first abdominal segment, of a perichitinous membrane structure, that's that membrane that's going to vibrate. there are synchronous muscles that move it. And so if you remember, I mentioned that last week, even if it wasn't locomotion, because it is a particular type of synchronous muscle that has a much higher frequency than other synchronous muscles used for locomotion. And it actually is able to operate at frequencies 2 to 3 times faster than other synchronous muscles. And it's partly due to large amount of those muscles is represented by the sarcoplasmic and sarcoplasmic reticulum. And so there is a lot of calcium released and recaptured by the sarcoplasmic reticulum, and that helps contract muscles faster. So this muscle is attached to the center of that membrane that's called the timbal, and the contraction and relaxation rapidly is what makes the noise, or that vibration, I should say, because it's not really noise. Tracheal air sacs are also involved. They're behind the timbal, and they can produce resonance, and they can also prevent other tissues to dampen the sound. There are other muscles involved as well that can increase that shape, the convex shape of the membrane, and it just amplifies that vibration. And it's pretty loud in the insect world. I'm not sure how much 100 decibel correspond to for us in terms of an equivalent noise. Maybe that's something we can look up after the lecture. And so this is useless if the female is not able to hear it. So the females have what is called the tympanum that serves as an ear that can receive this signal, and it can also distinguish male songs. And it's on the second abdominal segment. So now by sounds produced by, or vibration produced by stridulation, it is the rubbing of two body parts together, not vibration of the membrane. And I know sometimes it's confusing. Some people don't know the difference between the cicada membranes and between vibration and stridulation, basically. And so cicadas have a membrane, but crickets stridulate. And so basically, there's one body part that has teeth-like structures, and that is called a file or pars stridens. And an opposing part has a scraper that is, or plectrum, that is scraped on the file. And that's what produces the sound. In crickets, it's when the wings are closed. Then there is another structure that is called a harp. It's a corrugated area that vibrates through the stridulation. There's also a thin cuticular structure that's called the mirror that is supported by the wing veins, and it vibrates when the scraper and file are touching, or when the scraper scrapes the file. And these are a picture at the microscope of the different structures. So you have the file here. Well, I guess that's all there is. There's not another structure. I chose a wrong picture for that, but I mean, that's pretty nice still. If you ever wondered what it looked like close up. So now there's adaptations based on this mechanism. For example, the male mole crickets that also stridulates have found a way to turn their burrow into an amplification chamber, acoustical chamber, which increases the heart vibration by about 4%, which to us doesn't sound like a lot, but to them that makes all the difference. And so they use that to attract females. In beetles, there's also some examples of stridulation organs. Tachinid flies are able to pick up on stridulation of male crickets, and they are parasitoid flies, and so they use that to find hosts for their eggs. So they pick up the calls of the males, and then they fly near them and deposit their eggs either near them on the male crickets. And so some cricket populations have been able to evolve avoidance strategy by communicating silently. I don't know much about that, but I mean, that kind of illustrates how communication drives natural selection as well. And then in some Dipteras, for example, mosquitoes and Drosophila, during mating, they are able to produce sounds or different tones to attract mates based on wing frequencies. And wing frequency is also apparently used by honeybees to locate food or indicate the location of food. So then there's another type of acoustic communication that uses a substrate to produce a vibration. And a lot of insects produce that type of vibration using plants or other substrates. It could be different parts of the plants like the stems, leaves, roots. And it's definitely for short distance communication, less than 2 meters. And it's usually for courtship or to locate other individuals of the same species. But they can also be used as warning signals for predators. So 2 examples, the tok-tok beetle, African tok-tok beetle is able to tap the substrate with its abdomen, and termites also use that type of substrate tapping to determine the quality of the wood and also to generate alarm signals by drumming their head on wood. Yeah, and finally, the last type of acoustic communication that we know in insects, maybe there's more. It's through air expulsion. So a lot of you might be familiar with Madagascar hissing cockroaches. That's how they got their names, because they can hiss, and that's because they can expel the air through their tracheal system. And it could be because you're disturbing them or doing mating. So if you go near a colony, you're going to hear hissing no matter what. It could be just because you're there and they don't like it, or because they are trying to find a mate. And so it's because their spiracle on the 4th abdominal segment is modified, and it creates a shape that allows that loud sound to be produced. Then the tsetse fly is also able to produce sounds during feeding and mating through pushing air out of its tracheal system. Then we have tactile communication. So clearly that's for short range communication, because they need to be able to touch. And it's generally known for aggressive behavior or sexual encounters. For example, when crowded, aphids can sense that there are aphids close to them. And a lot of insects don't like being crowded, and so they can kick in response to that crowding. And so it physically displaces other aphids away from them. Tactile communication is also used to communicate with chemoreception. So that has aspects of touching, but also chemical communication. And for example, in ants and termites, antennal tapping is very important for the communication within the group. But yeah, we're gonna move on to chemical communication. Trying to stay relatively short to not bore you out of your mind. So chemical communication, what is it? It's just the emission of a chemical that is received by another insect, and so they can communicate with that. Insects use a lot of chemicals to communicate, and they're using those for mating, gathering, find food. Obviously, it smells like food that can go there. It's like you when you smell good cooking. Oh, what's going on? They can identify the nest mates, but also they can use it as a defense mechanism. So general chemicals are classified based on their function. Their function in the interactions that they mediate. So we have hormones, which are produced within an organism for use by this organism. So that's not what is involved in, or not directly involved in communication. What is involved in communication are semiochemicals, which are any chemicals mediating an interaction between two individuals. They could be the same or different species. And there's different types of semiochemicals used in communication in insects. The pheromones, you've probably heard that before. So that's for intraspecifics within a species, intraspecific communication, and allelochemicals for interspecific communication. So pheromones first, they are secreted from exocrine glands. So opposite to hormones that are secreted by endocrine glands. And I know you've already talked about that previously. So these glands, the exocrine glands that produce pheromones, are modified epidermal cells. What they produce and contain is usually in pretty small concentration. And the compounds that make up those pheromones are pretty common to a lot of organisms, and it's really the precise proportion, the blend that will confer the species specificity. There's two types of pheromones, with releaser pheromones and primer pheromones. The releaser, they stimulate an immediate and reversible behavioral response that is mediated through the nervous system of the receiver. In opposition, the primer pheromones mediate a physiological change by any system of the receiver. It could be nervous system, it could be something else. And one example is the queen bee pheromone that suppresses the worker's ovarian development. So it's not a nervous response, that's a reproductive system response, basically. Yeah, I'm not sure I'm going back to that. So the releaser pheromones, there are different categories. We have sex pheromones, aggregation pheromones, alarm pheromones, trail pheromones, dispersal pheromones. There's a lot of attracting a mate. So remember, it's within one species, so attracting a mate, but also attracting other individuals of your group to a same point for to signal them that there are food resources, or alerting them that there's a predator, so that would be alarm pheromones. These are a few examples. The synthesis occurs throughout the adult life, but it could be, I think I wrote hormones, but I meant pheromones. I think it could be mediated depending on circumstances, based on the environment and physiology of the individuals. And so I mentioned the exocrine glands that produce pheromones. There's two types, type 1 and type 2. And the type 1 are simple epidermal cells with a simple reservoir, epithelial reservoirs that stores the secretion. And the type 2 have both secretory and duct cells, so a little bit more complex. Yeah. So there's, here are two examples, female lepidopterans that can release pheromones from type 1 glands to be acquired by hair pencils of the males. And then in honeybees, where type 2 glands are used to produce alarm or sex pheromones. So like I mentioned, the basic components of pheromones can be common to different species, but it's their blend that really make the specificity. And they can disperse pretty fast, but that requires to use smaller molecules. And for more persistent smell of pheromones, they use larger molecules. So the activity is determined by their size, the molecular shape, and the functional groups and unsaturation of the molecule itself. And so, for example, in Ostrinia corn borers, there's 2 different species that use the same type of blend, but what changes here is one enzyme that will end up giving 2 completely different pheromones. So the genome of these 2 insects encodes for an enzyme that's called the desaturase that is used for the synthesis of pheromones. But in the European corn borer, there's one isoform that's active that's called the delta 11 desaturase. But in the Asian corn borer, it's the delta 14 desaturase. So there's just a slight change in isoform that will produce 2 completely different pheromones in the end. I'm trying to keep it short. So the pheromone synthesis is regulated hormonally. So there's an internal process that controls the external production of pheromones. So that's controlled by a neuropeptide that's called the pheromone biosynthesis activating neuropeptide, PBAN, or I call it PBAN. I don't know if other people call it that, but it's produced in the subesophageal ganglion. And so that's a small peptide, 33 to 34 amino acids, and it regulates pheromone production by involving signaling in the cell, basically. It can also, there's different forms of that little peptide, small peptide that controls different processes in the organism. Could be pheromone synthesis, but there's also non-communication processes like melanin synthesis or diapause or gut contraction. Trying to find the main message here so we can go a little bit faster. It just binds to a receptor on the membrane of the cell, and then it triggers a cascade of different events that end up being the production of a pheromone. And you'll have the PowerPoint, so if you want to go back to that. Yeah, so for example, the target of PBAN activity are glandular epithelial cells on the ovipositor tips of some species, for instance. And in the case where sexual pheromones are produced by the binding of PBAN, it is then inhibited after mating occurs. And since the subesophageal ganglion produces it, that's also what inhibits it. Yeah. Skip that. Okay, so primer pheromones. So in this case, I remind you the physiological change can be in the receiver, can be of any system, nervous system, or other, opposite to the releaser. So actually, these are a type of pheromones that have not been very well described because apparently they're very subtle effects, and also we don't really know how to study them. And one example, and I'm going to try to keep it short, is the primer pheromones are used a lot in social insects. And so one of them is the QMP, queen mandibular pheromone in honeybees, which is the one that I mentioned that inhibits the ovarian development of workers to maintain them as non-reproductive, because like most of you here already know that, but the queen is the only one that produces offspring in the hive. Okay, so now we have allelochemicals. So this is another type of semiochemical. Sorry, it kind of escaped my mind momentarily. So these are the chemicals that mediate the communication between 2 different species. And there's also different types of allelochemicals. So for instance, we have the allomones that are favorable to the emitter, but not the receiver. So for example, defense chemicals, there's a few examples. One that is, I don't know, kind of spectacular is in the Bombardier Beetle. They can discharge kinones to their attacker that are heated at 100 degrees Celsius. So it's boiling temperature. So that's an example of an allomeone. In that same group of allelochemicals, we have K-hormones, and these ones are adaptively favorable to the receiver, but not the emitter. So basically, these are allelochemicals that have evolutionarily backfired. For example, mosquitoes are able to find us as hosts because we produce carbon dioxide. It's not the main purpose, but it's just that we produce it and they are able to locate it and find us like that. They can also find sense lactic acid, which is another example of pheromone. Then varromites are attracted to honeybee drones because they produce fatty acids, same thing, fatty acid esters, sorry, and the same thing, they just produce fatty acid esters, it's not their fault, and varromites are attracted to that. Then a third type of allelochemical, cynomone, are allelochemicals that are adaptive favorably to both the receiver and the emitter. And so that's, I mentioned that very early in the presentation, the tripartite relationship communication between the herbivore that feeds on a plant and the plant calls for a parasitoid. That's what it is. So for example, the parasitoid wasp Trichogramma, parasitoid, I don't remember. But they're attracted to tomato plants because the tomato plants produce a cynomone that basically calls them. And so when they arrive at the site of the tomato plant, they can find a suitable host to parasitize. And bark beetles apparently also are able to use terpenes as pheromones to also attract, or the pine use that as pheromones, sorry, I got confused, to attract parasitoid wasps to parasitize bark beetles. Okay, you're gonna be relieved, it's the end. So these are a few, well, this is my main resource for references. And also I invite you to check out the links in the presentation. And this is just an overview of evolution of the lineages of insects that use light or sound for communication. And this is the paper that I selected. I'm not sure if you have read it yet, so I'm gonna let you lead what's happening right now before we end the session, because I think you're supposed to work in groups, right? Dependent on who's there. So there was a lot in this lecture. So there might be points that are not super clear, and I'm hoping that when you have the PowerPoint, you'll be able to go back to it. And if you have more targeted questions, you can ask myself or Troy about them. Any questions online? Okay. Ah. Oh, okay. No, that's good. Yeah, you don't need to have questions right now. We can digest the information and then get back to you. And if you don't have any group interactions.
I think we usually do those on Wednesday.
Ah, okay.
Yeah,
Okay.
some of the papers are like assigned, and we read them, and then we talk about them on
And then, yeah, we
Wednesdays.
talk about it on Wednesdays.
Okay. But don't you have two papers to discuss then on Wednesdays? Because you have lectures on Mondays and Wednesdays.
We normally only get one that like it's assigned on Wednesday and then we discuss it the following Wednesday.
Oh,
And then the next
Yeah,
one's
we
assigned.
get one per week and then we always just discuss it on Wednesday.
okay. Yeah, I haven't posted the one for next lecture, so I guess you wouldn't discuss it before next week, so it's fine. But yeah, we can discuss the one on different dialects and flies on Wednesday then.
Okay,
Okay.
Yep. I'll read it. But
It's somewhat, there's some points that are connected with the lecture that I'm gonna present on Wednesday.
Right, sounds good. I'll read it for them.
Okay, so I'm gonna stop sharing and the recording. I hope you have a good day.
thank you.
Yeah, thank
See
you.
you next Wednesday.